SNAB, where we aim to make A-level biology as simple as a cell. So today we're understanding the role of meiosis in ensuring genetic variation through a production of non-identical gametes as a consequence of independent assortment of chromosomes and crossing over of alleles between chromatids. So the meiosis is the process that produces cells that are genetically different from each other. So the cells are not identical, and gametes are produced through meiosis. So during meiosis, the parent cell goes through 2 rounds of division called meiosis 1 and meiosis 2. So what happens in meiosis 1? So the chromosomes replicate before meiosis 1 takes place, and during meiosis 1, the homologous pairs of chromosomes split up. So that produces 2 haploid nuclei. So now we're looking at meiosis 2. So a chromosome is made of 2 thread-like strands called chromatids. The chromatids in a chromosome split to produce 2 haploid nuclei. And this happens to both the haploid cells produced in meiosis 1. So now you basically have 4 haploid nuclei. So it helps to keep track. So each chromosome now contains a single chromatid each. So meiosis is quite advantageous compared to mitosis, as it allows genetic variation, and so that increases the possibility of having advantages through natural selection. So now we're looking at sister and non-sister chromatids. So non-sister chromatids are the 2 chromatids on different homologous chromosomes. So they are genetically distinct as they originate from the paternal and maternal chromosome. And sister chromatids, they are identical copies of a single replicated chromosome. So that's the distinction, and it's quite good to know so that the different processes you actually study make sense. So now we're looking at crossing over, which is something that you should most definitely know. And this is the process by which non-sister chromatids exchange alleles. And this takes place over meiosis 1. The homologous chromosomes pair up and are very close to each other, and the paired chromosomes are called bivalents. The non-sister chromatids can cross over, so emphasis on non-sister chromatids, so they can cross over, and the point where they cross over is called the chiasmata, and this crossing over results in a section of the chromatid breaking and rejoining the chromatid from the other chromosome. So basically swapping bits. That's how you can think about it. And this randomizes the combination of alleles further. So now we're looking at independent assortment, which is another whole process that can increase the variation in the cells produced. So this is the process that produces different combinations of alleles in the daughter cells. And this is the result of random alignment of homologous pairs along the equator of the spindle during meiosis 1. So the homologous chromosomes pair up and are pulled towards the equator of the spindle. So it's quite similar to what you would see in mitosis, almost. So after random rearrangement in the middle, the homologous chromosomes are separated by being pulled apart by the spindle fibers back to the poles. And you can calculate the number of possible different chromosome combinations using the formula 2 to the power n, where n is the number of chromosomes in a haploid cell. So that is all from Professor SNAB today. It's quite a detailed video, this one, so you might want to look back on it and see if things make sense, and if it doesn't, let me know and I'll get back to your questions. If you have any questions, let me in the comments below, and don't forget to like, share and subscribe.